Hello, this video is about cellular respiration and fermentation. We all hope that you are going to understand and experience more about these topics. Cellular respiration. Cellular respiration is a metabolic pathway having roles on breaking down glucose and producing ATP. During the process, the glucose molecule is broken down into carbon dioxide and water, and along the way, some ATP is produced directly in the reactions that transform glucose. And there are two types of cellular respiration, which are aerobic respiration and anaerobic respiration. Aerobic respiration. Aerobic respiration. Aerobic respiration is the process by which organisms use oxygen to turn fuels, such as fats and sugars, into chemical energy. Respiration is used by all cells to turn food into energy that can be used to power cellular processes. The product of respiration is a molecule called adenosine triphosphate or ATP, which uses the energy stored in its phosphate bonds to power chemical reactions. It is often referred to as a currency of the cell. Aerobic respiration is much more efficient and produces ATP much more quickly than anaerobic respiration. This is because oxygen is an excellent electron acceptor for the chemical reactions involved in generating ATP. 4 steps of aerobic respiration are glycolysis, preparatory reaction, Krebs cycle or citric acid cycle, and electron transport chain.
So the starting part of cellular respiration is glycolysis. Glycolysis takes place in cell cytoplasm. Glycolysis is the metabolic pathway that converts glucose into pyruvate by 10 enzymatic reactions. The step of glycolysis involves 10 enzymatic reactions, but we can classify it into 2 phases. That is energy investment phase and energy harvestment phase. The starting phase is energy investment phase. Start with 2 ATP used for activation energy to rearrange glucose. 2 activation energy rearranged by glucose CO2 compounds by attaching 2 phosphates from each ATP. The 6 carbon compounds split into 2 3 carbon compounds with a phosphate still attached. For the energy harvestment phase, the 2 3 carbon compounds is oxidized. It means that they lost electrons. Then they rearrange into another 3 carbon compounds. 2 NAD plus become reduced and gain electron to NADH. 2 NADH is produced. It will be sent into mitochondria to be used into the electron transport chain. When the NAD plus came in and they carry the electron, they also bring the phosphate with them to attach to each of the 3 carbon compounds. Each NAD plus got 1 phosphate, so total 2 NAD plus got 2 phosphates. Then 2 phosphates attach to the one of the 3 carbon compounds, and 2 more phosphates attach to another 3 carbon compounds. 4 ATP will produce by donating each of the 4 phosphates to ADP. The 2 3 carbon compounds then change into a different 2 3 carbon molecule called pyruvate. The result of the glycolysis is 2 pyruvate with 3 carbon.
Preparatory reaction. Preparatory reaction is the next step of glycolysis. It is also called pyruvate oxidation. It occurs in mitochondrial matrix. 3 carbon pyruvate enters and a carbon dioxide is removed, leaving a 2 carbon acetyl group. 2 carbon acetyl group binds to coenzyme A, producing acetyl-CoA that goes to the Krebs cycle. So, for the products of preparatory reactions, the inputs are 2 pyruvates and 2 NAD+, and the outputs are 2 acetyl-CoA, 2 NADH, and 2 carbon dioxide. So there's no net ATP and there's 2 net NADH.
The next cycle is Krebs cycle, which has another name is the citric acid cycle. This cycle occurs in the matrix of mitochondria. It's starting with acetyl-CoA or 2 carbon compounds that get from the previous cycle will attach to 4 carbon compounds and form 6 carbon compounds. Then 6 carbon compounds will oxidize. This causes 1 carbon molecule to remove and combine with oxygen. This will create carbon dioxide. Some energy in the forms of electrons will attach to NAD+. So NAD+ will receive electrons and change to NADH. In the third step is similar to the previous step. So the 5 carbon compounds will oxidize into 4 carbon compounds, and carbon molecules will combine with oxygen to create carbon dioxide. And then NAD+ will receive electrons and turn to NADH. This is similar to the second step. Next, 4 carbon compounds will rearrange itself and oxidize. This makes ADP will convert to the ATP. Then it will rearrange itself again to convert FAD to FADH2 by transferring 2 hydrogen atoms. For the last step is 4 carbon compounds will oxidize NAD+ to form NADH. And then 4 carbon compounds that left will wait for the next cycle. For the inputs of the Krebs cycle, there are 2 acetyl-CoA, 6 NAD+, 2 FAD, and 2 ADP. And for the outputs of the Krebs cycle, there are 2 ATP, 6 NADH, 2 FADH2, and 4 carbon dioxide. For ATP, it will further used by the cell. For both NADH and FADH2 will send to the electron transport chain and used to produce the ATP. For the carbon dioxide, it will leave the matrix of mitochondria and release to the atmosphere.
Next, we are going to talk about the electron transport chain. The electron transport chain was the process taking place in the inner membrane of mitochondria. It was a process that will make 32 ATP molecules. It was the last component of aerobic respiration. The input of the electron transport chain were NADH, ADP, FADH, and oxygen, and the output will be NAD+, ATP, FAD, and H2O. For the process part, the first NADH made from the other cycle will give the electron to the electron transport chain. As a result, NADH will oxidize into NAD+, as the same will happen to the FADH2. It will give electron to the electron transport chain and will oxidize into FAD. The electron from both NADH and FADH2 will move past the transport chain along the way. The energy made while the electron move will be used to pump hydrogen ion across the crystal into the intermembrane space. Later, once the electron releases most of its energy and reaches the end of the chain, it will combine with an oxygen molecule forming H2O. After the hydrogen ion has been pumped into the intermembrane space, it will form an electrochemical gradient. Once the chemical gradient is large enough, it will be ready to release, and then it will release the hydrogen plus through the ATP synthase, as it will create a kinetic energy that will be used to forming the ATP.
Anaerobic respiration. Anaerobic respiration. Anaerobic respiration is the process that transfers energy from glucose to cells. It occurs when oxygen is not present, and it transfers large amounts of energy quickly. Fermentation. Fermentation is a chemical process by which molecules, such as glucose, are broken down anaerobically. More broadly, fermentation is the foaming that occurs during the manufacture of wine and beer. a process at least 10,000 years old. The frothing results from the evolution of carbon dioxide gas, though this was not recognized until the 17th century. French chemist and microbiologist Louis Pasteur in the 19th century used the term fermentation in a narrow sense to describe the change brought about by yeast and other microorganisms growing in the absence of air anaerobically. He also recognized that ethyl alcohol and carbon dioxide are not the only products of fermentation. For instance, yeast performs fermentation to obtain energy by converting sugar into alcohol, and bacteria convert carbohydrates into lactic acid. The examples of the products of fermentations are beer, wine, yogurt, cheese, certain sour foods containing lactic acid, including sauerkraut, kimchi, and pepperoni, bread leavening by yeast, sewage treatment, some industrial alcohol production such as for biofuels, and hydrogen gas. For the alcoholic fermentation, yeast and certain bacteria perform alcoholic fermentation where pyruvate from glucose metabolisms is broken down into ethanol and carbon dioxide. The net chemical equation for the production of ethanol from glucose is C6H12O6 or glucose to C2H5OH or ethanol plus 2CO2 or carbon dioxide. Ethanol fermentation has used the production of beer, wine, and bread. It's worth noting that fermentation in the presence of high levels of pectin results in the production of small amount of methanol, which is toxic when consumed. And for the lactic acid fermentation, the pyruvate molecules from glucose metabolisms, glycolysis, may be fermented into lactic acid. Lactic acid fermentation is used to convert lactose into lactic acid in yogurt production. It also occurs in animal muscles when the tissue requires energy at a faster rate than oxygen can be supplied. The production of lactic acid from lactose and water may be summarized as C12H2012O11 lactose plus H2O or water to 4CH3CHOHCOOH lactic acid. For the similarities of aerobic and anaerobic respiration, both aerobic and anaerobic respirations are methods of generating energy. They also both start in the same way, with the process of glycolysis. Glycolysis literally means sugar splitting and involves breaking a sugar molecule down into two smaller molecules. In the process of glycolysis, 2 ATP molecules are consumed and 4 are produced. This results in a net gain of 2 ATP molecules produced for every sugar molecule broken down through glycolysis. This is where the similarities between aerobic and anaerobic respiration end. And lastly, for the differences between aerobic and anaerobic respiration. Aerobic respiration used for continue the ETC in a highly efficient process that ends up with 36 to 38 molecules of ATP. Cells that are deprived of oxygen but do not normally use anaerobic respiration may leave the end products of glycolysis sitting around, obtaining only 2 ATP per sugar molecule they split. Anaerobic respiration cell may continue the ETC to extract more energy from the end products of glycolysis.